Hello, welcome to Chapter 11, Fundamentals of the Nervous System and Nervous Tissue. This is the third in a series of 4 videos for Chapter 11. And today we're going to talk about synapses. And this should go pretty well because everybody's already learned about synapses when we did the neuromuscular junction back in Chapter 9. So in this cartoon, we'll talk about the synapse, and we're going to call this guy up here neuron number 1. We'll call this guy neuron number 2. So in neuron number 1 has a cell body, so that's this thing. So does neuron number 2. Neuron number 1 has dendrites, these projections of the cell membrane. So does neuron number 2. And then it's not listed on this cartoon, but there's this thing called the axon hillock. That's the junction between the cell body and the axon. Both neurons have an axon hillock. And then both neurons also have a myelin sheath. So I'd be really impressed if you can tell me if these are neurons from the PNS or CNS, and then tell me why you know what your answer is. If you said they're neurons from the PNS or peripheral nervous system, you'd be right, because these guys are single Schwann cells wrapped around the axon that make up the myelin sheath. So neuron number 1 is going to talk to neuron number 2 at this junction called a synapse. And when you take this square here and you enlarge it, so they enlarge it to a bigger cartoon here, you see that, well, what you've seen in Chapter 9 already. So this would be the axon of neuron number 1, and then this would be a dendrite of neuron number 2. And you can see the dendrite of neuron number 2 has a receptor for these molecules that neuron number 1 is going to exocytose or secrete. So some terminology: a synapse is a junction that mediates information transfer from one neuron to another, and the way you do that transfer is through these neurotransmitter molecules. So this axon terminal doesn't actually make direct contact with this dendrite on neuron number 2. So you have to use these neurotransmitter molecules to send a message from neuron number 1 to neuron number 2. So, and then this is the synapse, the space between neuron number 1 and neuron number 2. So neuron number 1 now is not going to be called neuron number 1 anymore. It's going to be called a presynaptic neuron. So it's the neuron that's pre, and if it's pre to the synapse, then neuron number 2 would be post to the synapse, or the postsynaptic neuron. So the presynaptic neuron conducts impulses toward the synapse. So, and we saw this in the neuromuscular junction. So the neuromuscular junction, that motor neuron axon, was going to conduct action potentials down the length of its axon. And then when the neuromuscular junction, if you remember, there were voltage-gated calcium channels in the membrane of the axon terminal. And when those voltage-gated calcium channels sensed a change in membrane potential, they opened, and if you remember, calcium rushed in, and it's hard to do this with the mouse, but anyway, calcium rushed into the axon terminal, and then a whole bunch of cool biochemistry happened, and the neurotransmitters in the vesicle fused to the axon terminal membrane here. It's the fusion event, and the neurotransmitter was released. In the Chapter 9, this would be the muscle cell, but in this chapter, this is a dendrite of neuron number 2. And neuron number 2, like I said, is the postsynaptic neuron, and it's going to transmit impulses away from the synapse. So what that means is, here's the dendrite of our postsynaptic neuron. Once it gets that signal from the neurotransmitter, it's going to transmit not action potentials, but graded potentials toward the cell body and then toward the axon hillock. And then it's a little more complicated than just this. There are molecules called NCAMs, so neural cell adhesion molecules, and for your nervous system, for your brain to get wired during your stay in the uterus, and then as your brain is wiring up until you're a young adult. So the average complete wiring time for a brain is 25 years for a human. You need these molecules called neural cellular adhesion molecules. So they're crucial for development of neural connections. Types of synapses. So there's a whole array of types of synapses. This first one that we're going to look at, it's called axodendritic. So here's our presynaptic neuron axon. Here's the axon terminals. And this part of the cartoon actually shows axosomatic, because the axon terminals are on the soma or cell body. These guys out here show an axodendritic synapse, because here's the axon of the presynaptic neuron, here's the axon terminals synapsing with dendrites of the postsynaptic neuron. So this big guy is the postsynaptic neuron. Axosomatic is between the axon of one neuron, the presynaptic neuron, and the soma or cell body of the postsynaptic neuron. So this one is axosomatic. This one over here is axodendritic. And then the third one is axonic. This is axon to axon. So this guy, this axon of this presynaptic neuron, has its axon terminals synapsing with the postsynaptic neuron axon to axon. So this space is actually the axon hillock. And then this is just a photograph, probably a scanning electron micrograph, colorized, of the axons of the presynaptic neuron synapsing on what I think is the cell body of the postsynaptic neuron. Okay, yes, it is. So yeah, that's right. So this yellow thing is the postsynaptic neuron. These guys are the axons of the presynaptic neuron in that scanning electron micrograph. So this is a cartoon. Like I said, we saw something like this in chapter 9. Only now we've got two neurons instead of a neuron and instead of a motor neuron and a muscle cell. So this is the presynaptic neuron. Here's the action potential in pink coming down the axon of the presynaptic neuron. These are voltage-gated calcium channels, these purple things here. When they sense the voltage change because of the action potential, then calcium will rush in, biochemistry happens, the neurotransmitter is released from the axon terminal, goes into the synaptic cleft, and it looks for a place to bind, a neurotransmitter binding site on the receptors of the postsynaptic membrane. So chemical synapse is specialized. So this whole area is the synapse. It's specialized for release of neurotransmitters and reception of neurotransmitters. So there's two parts, the axon terminal, the presynaptic neuron, and then the receptor region of the postsynaptic neuron that binds the neurotransmitter. And then you have the synaptic cleft. That's that fluid-filled space between the presynaptic and postsynaptic neurons. The transmission of the signal from the presynaptic neuron to the postsynaptic neuron is a chemical event because the neurotransmitter is a chemical, and that's how you communicate between the pre and postsynaptic neurons. This Mother Nature set it up this way because it ensures unidirectional communication between neurons, so that the communication is going from the presynaptic neuron to the postsynaptic neuron and the other way around. And this is just a more detailed cartoon of what we just talked about. So it tells you what I just said. The action potential comes down the axon terminal, voltage-gated calcium channels open, neurotransmitter is released by exocytosis, neurotransmitter diffuses across synaptic cleft and binds to receptors on the postsynaptic membrane. So here's a question. Up the top says the presynaptic neuron releases neurotransmitter in response to an influx of what kind of ions? So an influx just means movement into the cell. So movement out of the cell then would be efflux. But what kind of ions cause the release of this green ball, the neurotransmitter? If you said calcium ions, you'd be right, through voltage-gated calcium channels. This just shows cartoons a little more up close. Here's the postsynaptic neuron. And this would be the plasma membrane, actually, of the postsynaptic neuron. This would be the cytoplasm of the postsynaptic neuron. This would be this purple thing would be the ligand-gated channel. The green thing would be the neurotransmitter. So this, and then ion movement, it shows one arrow going in and one arrow going out. Which way do you think, which ion should I draw here as the ion going in? Would it be sodium or potassium? If you said sodium, you would be right. So that would make this potassium. So our neurotransmitter from the postsynaptic neuron, so this guy right here, here's postsynaptic neuron, here's the presynaptic neuron up top, here's the postsynaptic neuron. The neurotransmitter came from the presynaptic neuron, bound to this ligand-gated channel, and that caused sodium to move into the cell and potassium to move out. Sodium moves in more than potassium moves out, so that means the inside of the postsynaptic neuron becomes more positive. But here's the thing, this is a graded potential because this is a ligand-gated channel. Ligand-gated channels do graded potentials. Graded potentials don't get as positive as action potentials. So this graded potential, let's say the postsynaptic neuron started at -70. The graded potential may only bring the millivolts to go from -70 to maybe -60. So a little bit more positive, aka less negative. So in this cartoon, here's the presynaptic neuron, here's the plasma membrane of the presynaptic neuron, here's the postsynaptic neuron. When the signal is over, then the molecule, the neurotransmitter, has to be taken back up into the presynaptic neuron, and it's also degraded, so that the neurotransmitter, the green neurotransmitter, doesn't stay in this synaptic cleft and keep the signal going forever. Here's my cartoon that you've seen before if you watched the last video. So here's our major neuron that we're going to focus on. Here's the cell body. There's the axon. This area is the axon hillock. And here's the action potential being propagated down the axon. And then these green things, if you watched my last video, these are also neurons. I just drew them not as elaborate because it would take time, so I just did a quick sketch of a neuron. So these projections, green projections, are dendrites. Here's a cell body, here's an axon. And then in this cartoon it shows this guy right here would be the presynaptic neuron, and the major neuron that I drew would be the postsynaptic neuron. It just shows the neurotransmitter being released here from the presynaptic neuron, binding to a ligand-gated channel, and positive charges are flowing in. So remember, some potassium is flowing out, but mostly sodium is flowing in, so that's going to cause a change in the resting membrane potential from -70 to maybe around -60, because this is a local potential because we're using a ligand-gated channel. So this is a now they're also calling this a post-synaptic potential. So remember, here's the synapse. Here's the postsynaptic neuron, so they're calling this a postsynaptic potential. And they're saying it's a local potential. So it's local because it's not going to, it's a change in membrane potential that's not going to go very far. So remember, the action potential can travel the length of the axon. So it's a long distance potential, but this local potential, charges only diffuse a small distance, so they call it a local potential. And the strength of this local potential is determined by the amount of neurotransmitter released. So, and think about it, you could have many neurons synapsing. You could have many presynaptic neurons synapsing on this postsynaptic neuron, releasing a lot of neurotransmitter. So you could have a huge amount of neurotransmitter released. That means a whole bunch of sodium would flow in, and for sure it would get to the axon hillock and cause stimulation of these voltage-gated sodium channels, which would propagate an action potential. And then also the strength of this local potential inside the postsynaptic membrane would be determined by the time that this neurotransmitter stays in the synaptic cleft. So there's two types of postsynaptic potentials. You can be an excitatory postsynaptic potential, EPSP. That means positive charges are going to come in, and you're going to get a change in membrane potential towards the more positive end. And then if there's a excitatory, then there's going to be an inhibitory postsynaptic potential, and that's where you get more negative. So if you started at -70 millivolts, the resting membrane potential of our postsynaptic neuron, -70 millivolts. If you have an inhibitory postsynaptic potential, that means it's going to get even more negative. So it might go from -70 resting membrane potential to -80. And that's what these cartoons show, these graphs show. So let's look at this one first. If in our cartoon the positive charges came in, and you had let's say you have a giant electrode sticking in there. So that's an electrode, and it can read the membrane potential. So initially over here, before the stimulus, before the neurotransmitter bound to the channel, the resting membrane potential was -70. Now the neurotransmitter binds to the ligand-gated channel and positive charges flow in. This would be a excitatory postsynaptic potential, because look what happens over here on our graph. It gets less negative or more positive, but see, it only goes to about -60 millivolts. So it's still depolarizing. Sodium is rushing in here. That's a depolarization event right here. But this is a graded potential as opposed to our action potential. Remember our action potential, you start over here at -70, you get this slow depolarization phase, but then if the membrane hits -55 millivolts, that's the threshold potential, then you can fire an action potential. I should probably put +30 up here, because that's where the peak goes to. Then, if instead of positive charges, your neurotransmitter here stimulated this ligand-gated channel and negative chloride ion poured in. So negative chloride comes into the cell. That's a bunch of negative charges. Then your graph would look like this. So again, you start at minus 70 millivolts. Negative chlorine or chloride ion, starts to move into the cell, and you get even more negative. So it's a hyperpolarizing graded potential because you're even lower than the resting membrane potential. So local potentials then, so remember the local potentials are going to occur in the cell body, not the axon. They can be excitable or inhibitory depending on the neurotransmitter and the type of ion that goes into the cell. If the local potential is excitable, that means positive charges came in, and the graph looks like this. If they're inhibitory, that means chloride channels were open and negative charges moved in, and the graph looks like this. Local potentials are graded. That means they're proportional to the stimulus strength. So if you have 50 neurons releasing their neurotransmitter onto the postsynaptic cell or neuron, then a whole bunch of positive charges are going to come in, and this graph might go up to, I don't know, maybe minus 50. So graded local potentials are graded proportional to the stimulus strength, and then local potentials are also reversible. So if now the neurotransmitter is gone, that means the stimulus stops, and you'll go back to resting membrane potential, which is minus 70 in both cases of these graphs. Local potentials are decremental. That means the potential will decrease or get weaker as it travels from the point of stimulus. So the positive charges are here, they're coming in here. They've got to travel this distance to the axon hillock to try to activate these voltage-gated sodium and potassium channels. But as these charges move, the potential, the positive potential gets weaker because the signal is decremental. So now this is kind of, we talked about this, but let's go through it step by step. So the neurotransmitter from the presynaptic neuron binds to the chemically or ligand-gated channels. Sodium flows in and some potassium flows out. But the sodium influx into the cell is greater than the potassium efflux out of the cell. So you get a short distance, a net depolarization. So it looks like something like this. You get a net depolarization because the positive coming in is greater than the positive going out. So the net result is that you get a depolarization of the membrane. If enough charges come over here, if enough charges move through the cell body over to the axon hillock, enough of these positive charges come to this area, the axon hillock, then that means the membrane potential at the axon hillock, if it drifts up to minus 55 millivolts, which is getting more positive, right, or less negative, then you can fire an action potential. Then the action potential will travel all the way down the axon without changing its height. So the action potential will look the same all the way down the axon. And then I just said that if the excitatory postsynaptic potential is at threshold potential, that's minus 55. And if it gets to be minus 55 at the axon hillock, then those voltage-gated channels, sodium and potassium, will open and you'll get an action potential. And this cartoon just says a single excitatory postsynaptic potential results in a depolarization without an action potential being generated. So that's a single excitatory postsynaptic potential. But if you have a whole lot of them, then you can get an action potential. And that's the slide we talked about. If, on the other hand, a neurotransmitter binds to, let's say this is a chloride ion channel, you can get an inhibitory postsynaptic potential. So you'll get a hyperpolarization, so the inner surface becomes even more negative than -70. And if you get this inhibitory postsynaptic potential, then that's going to reduce the neuron's ability to fire an action potential. So let's say you have one neuron sending a positive signal, but let's say you have 3 neurons sending a signal for chloride to flow in, then that means the 3 neurons and their channels are opening channels for chloride. You'll have more negative charges in than positive charges, and you won't fire an action potential. And this just shows the cartoon that was in the book. So summation, this integration. If you are a postsynaptic neuron, how do you sum all the signals that you get? So this just shows in this cartoon a single excitatory postsynaptic potential cannot produce an action potential because it doesn't get the membrane potential at the axon hillock to -55, the threshold potential. But these excitatory postsynaptic potentials from the presynaptic neuron to the postsynaptic neuron on a dendrite can summate to reach threshold. So here's an initial excitatory postsynaptic potential, and then before it can go back down to the resting membrane potential, another excitatory postsynaptic potential fires. You get to threshold -55 millivolts, and then you can fire an action potential down the length of the axon. But remember, don't get confused on this. The graded potentials, the local potentials can only happen in the cell body. The action potentials can only begin at the axon hillock and then fire down the axon. IPSPs can also summate with EPSPs, and that's what I said in the previous slide. So if you get a signal, let's say this is a signal that will allow positive charges to come in. This is a signal that will allow negative charges to come in to the postsynaptic neuron. You have 10 positive charges coming in here, 10 negative charges coming in here. They cancel each other out, and you don't get a firing of the action potential beginning at the axon hillock. So this is called temporal summation that we just looked at. 2 excitatory stimuli close in time cause an excitatory postsynaptic potential, and they add together. So in temporal summation, you can have 1 or more presynaptic neurons. That's this guy, this green thing up here. That's the presynaptic neuron. Transmit impulses in rapid fire order. So you could have the action. You can have action potentials coming down at a high frequency in this presynaptic neuron axon terminal, or you could have another presynaptic neuron firing action potentials. So the point is that you can sum up these signals over time. So if all the potentials combined, so all these graded potentials combined, result in a threshold depolarization, then the action potential will fire. So no matter how many signals these dendrites of our postsynaptic neuron are getting, so let's say each one of these is getting some kind of signal for positive charges or negative charges to come in. Maybe the cell body is getting a signal for positive or negative charges to come in. All the potentials combined, all these local potentials from these ligand-gated channels combined, if it's enough to get a positive charge over here at the axon hillock and reach minus 55 threshold potential, then you will fire an action potential and it'll travel down the length of your axon. So this cartoon just shows here in the key, you have a green excitatory synapse number 1. Where's our teal? Okay, yeah, this just, for this cartoon, it's just the excitatory synapse. And it shows no summation here, so you have 2 stimuli, but you don't reach threshold potential. Here in temporal summation, you had your first stimuli, didn't reach threshold potential, but you had another stimulus come along before this could return to resting membrane potential, and that was enough to get the axon hillock to minus 55 millivolts, and you fire an action potential. So in spatial summation, the postsynaptic neuron, so here's our postsynaptic neuron, is stimulated by a large number of axon terminals at the same time. So it's just showing here 2 axon terminals are stimulating our postsynaptic neuron at the same time. And it says if all the potentials combined result in a threshold depolarization, then an action potential will fire. So whatever these stimulations are, if they're allowing for negative or positive charges to come in, if you can get the axon hillock to minus 55, that's threshold potential, and you will fire an action potential down the length of the axon. Okay, and then this cartoon just shows spatial summation, 2 simultaneous stimuli at different locations cause EPSPs that add together. So, and that's with this cartoon. So you have, here's a stimulus, so the neurotransmitter, basically. It's happening at the same time, so it's going to cause enough positive charges to get over here to the axon hillock, and you'll fire an action potential. In this cartoon, it says spatial summation of an EPSP and an IPSP. So here's the inhibitory postsynaptic potential, here's the excitatory, and they sum together and they canceled each other out, and you didn't get any change of membrane potential at the axon hillock. So repeated use of these synapses increases the efficiency of neurotransmission, and it also increases the calcium concentration in the postsynaptic neuron. Oh, and this is kind of interesting. So this is our cartoon. It shows, so here's a presynaptic neuron stimulating the postsynaptic neuron on the cell body. Here's a presynaptic neuron stimulating the postsynaptic neuron on a dendrite, and here's a presynaptic neuron axon stimulating the postsynaptic neuron on the axon. So it says release of an excitatory neurotransmitter, so that means you're going to get a depolarization by 1 neuron. So let's say this guy released a neurotransmitter and caused positive charges to come in, may be inhibited by the activity of another neuron via an axo-axonic stimulus. So you could have your positive sodium coming in here, and sodium is diffusing over here to the axon hillock. However, this axo-axonic synapse is making the region more negative, so you won't have you won't be able to fire